Okay, so I'm going to be talking as I was introduced. So thank you very much, Shri. So I'm going to be talking about grow your. I hope my title is not too misleading, but I will be talking about how we can grow kind of parts of a cornea. So you had to have a snazzy title, so that's what that is. So grow your own cornea: the do's and don'ts in stem cells in the eye. So I think with stem cells in general, there's kind of like a lot of hype. So I'll kind of address sort of what is possible, specifically in the field of the cornea and what we can do and what has been done, sort of thing. So I'll start off with a very easy question. Is everything okay with the PowerPoint? I hope it's okay.
Yeah, looks great.
Good, great. So a very easy question is, how do we see? And you might think, okay, that's an easy question. Easy answer, I know that one. But it's actually a lot of different things are going on in order to get an image in your brain. So obviously you're perceiving something, you're seeing reflected light off an object. This light has to pass through lots of different structures before it actually even reaches your brain. And all of these structures have to be intact. They have to be healthy. Otherwise, you're not going to be able to see the image as well as you could. So as you can see, it passes first through the cornea. So what the cornea does is it refracts the light. So it angles the light towards the lens through the iris, or through the pupil rather. So the iris controls the amount of light that actually is passing through, and then the lens focuses the light on a specific area of the retina in order to get you the clearest vision. And then when you reach the back of the eye, so you can see, I don't know, I'm pointing at my screen, I need to be pointing with my arrow. So as you look on at the retina, so these will then generate electrical signal from the light, and this will be passed along the optic nerve and then finally processed by your visual cortex. So if you have any injuries or anything goes wrong in any part of this pathway, you will have visual issues. But I'm going to be focusing mainly on the cornea, which is this very clear curved structure at the front of the eye. So most people focus on the retina because it's a bit more exciting, a bit more sexy, but we focus on the cornea because it's equally as important for clear vision. So this is this structure here at the front of the eye. So the cornea, as I said, is clear and curved, and it's consisting of 5 layers of different types of cells and structures. So I'm particularly interested in this top layer. So this is what we call the epithelium. And then below that, you have what's called the corneal stroma. And if you can picture that, it's lots of different overlapping layers of collagen fibers, and they're arranged in a very, very specific way in order to allow the maximum amount of light to pass through. Obviously, you need all the light to be passing through, but this minimizes any light that is absorbed by the way the collagen's arranged. And then you also have a couple of other layers, which I'm not going to talk about too much. So I'm going to focus on the epithelial layer. So as you can imagine, as I mentioned before, the cornea angles light towards the pupil, and the epithelium, which is what I'm interested in, this protects the eye from diseases and damage. And if you think about your eye, which part of it is constantly in contact with the outside world, it is this epithelial layer. So this is the layer that's constantly being barraged with UV light, microbes, and chemicals, etc. So this needs to be regenerated constantly, and it's constantly lost. Every time you blink, you'll lose some epithelial cells. And it is regenerated from these structures called the limbus. So this is kind of a ring all the way around the cornea. And in this structure, we have stem cells, so what we call adult stem cells. And I'll talk a little bit more about the different types that we have. So if you ever get damage to your epithelium, and particularly in this limbus region, then you will lose the stem cells that regenerate this epithelial layer. And I'm not sure when the kind of gory picture is going to come up, but if you're squeamish, I recommend you look away in the next couple of seconds. So if you get damage to your limbus, you can get what's called conjunctivalization, so where the whites of your eye, the sclera or the conjunctiva, will grow over the cornea, and you lose that clear structure, and light can't pass through, and you lose your vision. So this is usually where you get severe damage to your stem cells, and there's different things that cause this. But interestingly, one injury that can cause this is the acid attack. And in the UK, acid attacks have risen 250% in the last 5 years, which is a very worrying statistic. So this isn't the only thing that you would need that can cause this disease. You can also get limbal stem cell deficiency, where the stem cells in the limbus are lost. So this can actually be from contact lens overwear, or also just physical damage or other chemical damage. So how do we treat this condition, which causes very painful and also causes sight loss? So the best treatment is transplantation. However, there's only one cornea for every 70 needed. So this is not just in the UK, this is kind of worldwide. Different countries have different needs for transplantable material, but in the UK, corneas are very hard to get, and you'll be on the waiting list for quite a while. So what other therapies are there? And obviously this is where we turn to stem cells. So there are various things that people have looked at, but with stem cells, I'm just showing you some of these headlines here. There's a lot of hype about stem cells, and I think a lot of people think they are the be-all and end-all of curative options for cell therapies, etc. But I kind of want to just rein it in and say, what actually are stem cells? So you can't just put a stem cell in and it magically heals something depending on the stem cell. So I'll hopefully explain to you the different types of stem cells. And whilst they could potentially cure any disease, there's a lot of work that goes into getting them actually ready for transplant. So what can we do and what can't we do? So this is just a variety of studies. So this is just specifically for cornea. So don't worry too much about all the different acronyms, but the two most interesting ones that I'm going to be comparing are these are procedures to replace the stem cells that are lost. So when you get that damage in the stem cells in that specific area, the limbus, as I mentioned, when you get damage there, you will get conjunctivization and then resultant clouded vision. So these two surgeries, so this is cultured limbal epithelial transplantation. Basically, you take a little chunk of healthy limbus, so you've got a chunk of stem cells, and then you put it into your injured eye. So this kind of is counting on you having one healthy eye and then one injured eye. So this was a pioneering treatment, and it worked. It worked really, really well. So they just used adult stem cells that were already there and put them in, and they regenerated the cornea. However, this is quite a intrusive surgery because you're actually injuring the other eye to cure the other one. So a lot more therapies have come along. So all of these are, apart from these two, I think, are stem cell therapies. So this study is this is produced by a group in Japan. I won't go into the finer details of how we make these, how we regrow the stem cells, but essentially they have come up with a method to make them from what we call iPSCs, induced pluripotent stem cells, which I'll explain in a second. So there are two main types of stem cells. So you have pluripotent and multipotent or unipotent. So pluripotent stem cells can become almost any type of cell. So these are cells, they don't exist in the adult body typically, except when you beget a fertilized embryo. So an embryo is totipotent, essentially, and embryonic stem cells are pluripotent stem cells. So obviously, if you picture an embryo, these cells are going to have to become every type of cell in the body, so they have to be pluripotent. However, multipotent and unipotent stem cells, as you would think, so potent means they're potential, so they're multipotent and unipotent, they can only become certain types of cells or regenerate themselves, for example. So they're restricted to specific cell types, and they're often found in very, very specific niches, what we call niches or parts of the body. So as I've mentioned, the edge of the cornea, the limbus. So why we're interested in stem cells is because they can divide and renew themselves for long periods, sometimes indefinitely. They can give rise to other types of cells, and they can remain unspecialized. So they could potentially form other cells that are necessary. But I'm focusing on what induced pluripotent stem cells. So these cells can be produced from any adult cells, usually skin. So here I've got a little diagram here. Don't worry about the details too much. There's two ways. You could get just a regular donor. They could donate some of their skin, for example. You just take a biopsy, and then you can reprogramming these cells. So you can make them like embryonic stem cells so that they can then become any type of cell again. And you use a complex set of factors. So don't worry about the names again, but this is you throw these factors on. So these are genetic factors, and they will change the cell's very being. So they will, at a genetic level, they will instantly turn the cell from whatever it was before, a skin, fibroblast, for example, and then it will become this pluripotent stem cell that can become any type of cell. So you could take this from a donor, or you could actually take it from your patient and then reprogram the patient's cells, form these pluripotent stem cells, and then what you can do then is differentiate them. So you can differentiate the cells, and then to whatever cell type you want in theory, and then place them back into the patient, and then it's the patient's own cells being put back in. Which all sounds very good in theory, but it's very, very difficult to reprogram cells from a patient, for example, and also for this differentiation. There's a lot of complex factors that produce this effect. So I'm going to talk about now. So as I said about our need is we want to make these limbal stem cells, so the corneal epithelial stem cells. So how do we get from these iPSCs, which we can make, into the corneal epithelial surface cells? So there's a group in Japan that have developed a protocol to do this. It's essentially a 2D structure where you give certain amounts of factors and the cells will self-organize into different cell sheets, essentially, and you can then pick off the ones that look like corneal epithelium and then grow them up, and they will eventually have some stem cells in there, and that's how that works. And they have actually transplanted this into somebody, and it restored their vision. But as I said, you need to know how to program your iPSCs. So this group in Japan that we're working with, they've accounted for basically everything. So they've got the environment, the soluble proteins that will differentiate the cells, the pharmaceutical products that you can put in, the oxygen and carbon dioxide concentration. And what we're really looking at now is really fine-tuning these cells so that we can give them the environment they want. And we can do this by the extracellular matrix. So this is the protein meshwork in which all cells in your body are embedded. So if you think, for example, connective tissue, which the cornea kind of is connective tissue, but you can think cartilage as well. So the extracellular matrix is all of that meshwork that holds the cells in place. And we're particularly interested in one specific molecule in there, and they're called proteoglycans. So if you think of them, they kind of look like a bottle brush. So if you picture a bottle brush, you've got this metal chain with lots of kind of bristles attached. And the metal chain is the protein, and then the white bristles are these sugar chains that are attached to it. So actually it looks like this. But you can get lots of these side chains, and these basically can be read like a barcode, and you can get specific barcodes in different types of the body, and we think that they will have different effects. And they've often been thought as kind of space filler molecules, the extracellular matrix, and particularly these proteoglycans, these bottle brush-like structures. So here we have, they often just thought, as you can see, just space fillers. You've got our cat here, the cell, just with all these molecules sitting around, not really doing anything. However, recent research has shown that they're actually important in maintaining stem cells. And so it's less of kind of a random structure, and it's more like this. You have a stem cell with this very cute niche built around it that keeps the stem cell as a stem cell. And we think this is what's being lost when you get that conjunctivization. So if we can provide this to our pluripotent stem cells, will they form the natural stem cells better? So if we know what's there, we can provide them to the pluripotent stem cells. So if we look, we can see this is kind of a zoomed in molecular image of the cellular image. We can zoom in and see this is where our stem cells are in the cornea. So we can see what's here, and we can see if there are any of these barcoded proteins that we can find and then extract. And then we can then take those and hopefully recreate the environment that makes these stem cells to encourage them to differentiate more quickly. So this is an image here. So if you think of this is kind of where that target was on this previous screen. So where this arrow is pointing. So this is where all the stem cells are. They'll all be in this kind of dip at the bottom. And we found one specific barcode that seems to be very, very important in this area. So nobody else has ever looked at these types of molecules in this context. So this is what we want to focus on. So we want to get this specific barcode and take it out and have a look. So that's what I'm doing at the moment. So we want to see if we can take these glycocodes, we call them, so this particular barcode protein, and then we want to use it to grow from the differentiation that I talked about. Can we differentiate these pluripotent cells using this molecule, and does it improve the differentiation? So that's pretty much my talk. I hope that was straightforward. So I'd just like to thank everybody, my supervisors and other members of my research group and my funders as well. So I hope that was a little insight about my project.